Hello. Today we're going to talk about meiosis. Meiosis is that part of the reproductive system in which gametes, eggs, or sperm are formed. Previously, we have been talking about mitosis, and mitosis basically is a somatic cell that is, we've talked about, is 2N, or it has two sets of chromosomes that divides, and a parent cell that is 2N produces two cells that are identical. Now, that happens in every cell except those that produce sex cells, eggs and sperm. Egg, another name for egg is ovum. So in meiosis, you do start with a 2N cell, but that cell goes over two cell divisions. In these two cell divisions, that you produce four cells with one set of chromosomes, or 1N. These four cells are called gametes, and it could be eggs or it could be sperm. Now, in this PowerPoint, we are going to be talking about the details of how meiosis happens. And obviously, we all know that each of the children have some characteristics of the parents. But now, meiosis does two things, like you see over here. You take a cell that is a diploid cell, or two sets of chromosomes, and it makes a haploid cell, or one set of chromosomes that we call 2N is two sets, and 1N is one set. And this is a very good idea, like we have discussed in the past, because you're going to join an egg with a sperm, and you do not want to have 92 chromosomes in a human. You want to have 23 plus 23 is 46 chromosomes in that one cell. And how does this happen? Well, in this case, what happens first is that the homologous chromosomes will duplicate and make sister chromatids, like we all know it happens from mitosis. The sister chromatids are made during synthesis, okay, or S in the interphase. Now, different from mitosis, in meiosis, you see these homologous pairs. These homologous pairs will line up next to each other. Okay? And when these homologous pairs line up right side by side, then a few things happen. And one of them is this site of genetic recombination. So the homologous pair, one from mom, and the other one that is exactly the same from dad, they are right next to each other. They exchange genetic information. They switch. Chunks of DNA from chromosome from mom goes to chromosome from dad. And this happens, this is what is called genetic recombination, and it is basically the key to give us gametes that are different from each other. Okay? This happens in prophase, prophase 1 and metaphase 1 of the very first cell division. Okay? When this happens, then you're now seeing that this chromosome, the black chromosome over here, is not the same as the one we started with. It has a little chunk of a red. It has a little chunk of red over here that came from chromosome red. And this one has a little red chromosome over here that came from this one. Now you can see, different from mitosis, each of these sister chromatids are different. Okay? And this is what we call the first division of meiosis, in which homologous pairs line up, exchange or genetic recombination happens, and this happens quickly, and we then start meiosis 2. In meiosis, the scramble then of the genes in each one of the sister chromatids happen and give us then unique sex cells. Okay, and this is what we refer to when we say that in meiosis we create chromosomes that are different, giving us genetic diversity. So crossover happens, but this other concept of independent assortment happens too. We'll see that in a minute. Just a brief review. In here, you can see that there is a pair of homologous chromosomes. Chromosome 1 from mom, chromosome 1 from dad, chromosome 2 from mom, chromosome 2 from dad. And as we have said before, each one of these chromosomes have a gene that codes for the same trait. Like in here, you can see this gene codes for the color of the eye. So you could have brown eyes from mom, green eyes from... So in here, you can have the different color of the eyes, as well as you can have if you're tall or short. In this case, you can see that there's, as I said before, different genes in one chromosome. These are homologous chromosomes, but the trait can be different, right? The color of the eye or how tall or short you are is what makes it unique. We have been talking about ways in which these gametes are different from each other, and that happens because of this concept, two things, this concept called independent assortment. In independent assortment, you can have a chromosome like you see over here, in which they can line up red on top and yellow on the bottom, but you can also have it by chance for them to just lined up yellow on top and red on the bottom, making at the end of this process of meiosis 2, making a cell that has the red or the yellow or the yellow or the red, right? It depends. The other thing that makes it unique in terms of independent assortment is that this actual sister chromatid, this sister can line up on the top and this sister in the bottom, like you see over there, but you also have this sister, the bottom sister can line up on the top and vice versa. So that will make a difference, the way they line up, either red on top and yellow on the bottom, or the actual red sister, the sister chromatid on the top, can go on the bottom, and so on. In here, you can see that in independent assortment, it produces 2n distinct gametes, where n is the number of chromosomes. So in humans, n equals 23. 2 times 23 is 223, or 8 million different gametes that you can have in just one person, okay? And these are some of the examples of unique gametes, right? Not that you have this many, but a combination of 4 of these is what you could have if you were going to only have 3 chromosomes, okay? In this slide, you can see that there are 2 homologous chromosomes. These have gone through synthesis, right? So you see sister chromatids and sister chromatids, and they are lined up one next to each other. Now you know this is meiosis because they are one next to each other. When they are here in prophase 1 of meiosis 1, they are able to interact, right? These sister chromatids, this one can tangle up with this one, or this one can tangle up with this one. Here's just one example, and it's called a tetrad because a tetrad means 4 legs, 1, 2, 3, 4. They tangle up, and they literally, like I said before, chunks of one sister chromatid goes to another sister chromatid. Now, each of these chromatids are different from what you started before. And here you may not see a blue, but you could have the same blue chunks on red or red on blue. Crossing over, independent assortment and crossing over are what makes different types of gametes, eggs or sperm. Remember, gametes are eggs or sperm. This is also what generates genetic diversity. In here, you see a family that in here you see the whole process of meiosis 1 and meiosis 2. You start over here at the very top with 2 chromosomes, okay, one red and one black in here. These chromosomes go through interphase and synthesis occurs. Now you have sister chromatids. And now in PMAT1 of the first division, you have homologous pairs that goes through crossing over, and they line up next to each other, and they could be left red, right black, or vice versa, right? So the independent assortment happens in prophase 1, and crossing over happens between prophase 1 and meiosis 1. Now you have metaphase 1, anaphase 1, and then you're going to have telophase 1, and then cytokinesis 1. You form two cells very quickly, these 2 cells, because now all this synthesis doesn't have to happen the second time because the sister chromatids are already there. Okay, so this transition from cytokinesis 1 and goes directly to prophase 2, and then PMAT, prophase 2, metaphase, anaphase, and telophase 2, where just like in mitosis, sister chromatids are the ones that are going to be separated in telophase over here, forming 1, 2, 3, 4 cells. Note, you start with 2 chromosomes in this example, you end with 1 chromosome in each one of these cells, which shows you that you go to half of the genetic material in the process of meiosis. Here you are seeing the difference between mitosis and metaphase 1, right? In the first division, before the cell divides, you see that it is 2N. They both have 2 sets of chromosomes. But in here, this is chromosome 1, this is chromosome 1. These are homologous chromosomes. They line up one on top of the other, forming one row of chromosomes. When I show you a picture, I want you to notice that, because if you see that, these are one row of two chromosomes of each type, right? 2 blues and 2 reds in here, or 2 long and 2 shorts that are homologous pairs, but they go line up on top of each other. If it is prophase 1 in meiosis, you see that the homologous chromosomes are next to each other and form 2 rows of homologous pairs. Okay? When they line up over here, we were just talking about independent assortment happens, and this blue landed in the left, but it could easily land on the right, and vice versa over here. These sister chromatids can line up with this leg over here, or it could line up with this leg on the other side. So independent assortment is part of that genetic diversity that in the end will give you different sister chromatids in each one of the cells. Okay, so that's that lining up. And again, I'm going to write it here. This is in prophase, in mitosis, and this is prophase 1 or metaphase 1 of meiosis, of the first division. If you don't have the number 1 in there, you will not get them right in your assessment. Here you have this picture of a real chromosome in here, in which you can see that the X chromosome is a lot larger than the Y chromosome, and this is in the condensed form of the chromosome. Okay, it's not like a loose string anymore. Also, you see that it's like the letter X is doubled, right? You have these 2 sister chromatids, so you know that synthesis already happened. In here, you can see that you have it is basically showing you what happens inside the germ cell is 2x